So I did my PhD here in Barcelona with Gustavo Deco, and now I'm in Portugal as a postdoc, an independent position. And my main project is to investigate pathoconnectomics and how to rebalance brain activity, let's say. But this is a modeling work, so I will start to introduce you in this quest for the brain's constitutive equations and about brain rhythms, how to go from local activity to global, what is understood by metastable synchronization, collective frequencies, damped oscillations, then what are these metastable oscillatory modes that emerge both in real data and in models, and we'll discuss briefly. So when we have two physical quantities, anything can be forces, fields, matter, systems, and we want to see how the reaction of B, so how the action of A produces a reaction on B, we need to look for the equations that can explain these mechanisms. So for example, this is if we, this forms the system, or it can induce more nonlinear phenomena, such as it can start vibrating, it can change its color, so we don't know what is happening. And this is called, to solve these problems in physics, to look for the constitutive relations or the constitutive equations between things. And examples, for example, are the fluid flows in response to pressure waves, liquid crystals in response to electromagnetic fields, but also Maxwell's equations, electromagnetic equations are constitutive relations between the magnetic field, flexoelectricity, electroelasticity, magnetohydrodynamics. So there are different examples of constitutive equations that are needed to really understand the system. And maybe, so we are not considering these things in the brain, but they may be occurring as well. So there are many interesting phenomena occurring. And so these equations can be linear or non-linear, which are the most interesting ones. They can be reversible or not. They can be reversible, as Gustavo explained as well, and reciprocal in the sense that the action of B also can provoke an action on A. So to really understand the system, we need to unveil its constitutive equations. So this is just a general overview. So on the brain, we know and we are seeing that the brain activity reacts non-linearly to transcranial electric or magnetic stimulation. So there is a reaction from the system, from an action from something else. And this reaction is found to relate to consciousness levels. So I already know basically the works from Massimini, but others as well. But most importantly, I need to point in this direction, right? Most importantly, this reaction seems to offer therapeutic effects, and that you know better than me, but I believe you that there is something in this reaction that is therapeutic. But why and how this occurs remains unclear, and this slows the development of therapeutic strategies. So to understand the system, let's say, we first observe, we then explain, predict, control. So observe, we've been observing brain activity for 100 years now with different types of recording techniques since already the 19th century recordings, first recordings on animals, then we've developed really amazing tools to see the brain with ever more precision. But still, we need to explain. So to explain what we see, we try to develop models. So we think of what could be giving origin to these patterns that we see. And then if these models are good enough, but we are still like the colors, green is how much we have achieved so far. Now at the prediction level, it's still a bit more phenomenological. So in a way, we need to compute the consequences of our explanatory models to validate with experiments. And then ideally, once we understand everything, we will be able to control. And when we understand everything, we can make predictive algorithms to design personalized strategies to improve the treatment success, okay? So I'm at the explanatory level. I think we need to improve at the explanatory level in order to evolve. And one of the things that I find really intriguing is brain rhythms. What I want to try to understand or find explanations for this. These brain rhythms have been, so they are not questionable, let's say. So since the first recordings with galvanometers, you could see the emergence of alpha waves. So when the cats opened their eyes, this Richard Caton described that the alpha waves went down and you'd see an increasing in beta waves. But this was with the pencils writing on a paper, right? And you had to measure the periodicity with a ruler. But so they are there, clearly there. Their power is modulated over time. Their frequency power relates to behavior, consciousness, different types of conditions, let's say. There are multiple characteristic spatial patterns. This is also important. They are very macroscopic, the way they're organized. And they are used widely for brain-computer interfaces at different levels for neurofeedback, but also for control of machines and things. But still, how and why they emerge remains under debate, right? So at the neuronal level, so when we Google what is the origin of EEG activity, okay, so it's the main theories that, of course, so this is generated by the concentration of ions according to the polarization of the membrane of the neurons, right? It was explained already. So you could have a negative signal in the scalp, depending on the organization of the ions along the pyramidal neurons. And but still, for these signals to be detected, it is accepted that it requires a very large number of neurons firing synchronously and in parallel to be detected in EEG. But still, okay, let's say, okay, we accept this. But even then, the theory fails to explain why there is a frequency modulation, right? Well, it's not that it fails, it just doesn't address it even, why the frequency is modulated and why the patterns are organized at the microscopic scale. So what coordinates the system at the global scale in this frequency domain and spatial domain is unclear. So here, I'll go for a degree of reduction compared to Javier's talk was more in the neuronal level and Enrique. This is a reduction. I'll take what are the neuronal populations or mass of neurons. They are known, they have this amazing property that if you just take a piece of cortex and you put it in vitro in a tube, with the cholinergic excitation, activation, and tonic excitation, you can induce gamma oscillation. So it's like an excitable tissue, a biological tissue that when you have certain concentrations in the extracellular milieu, it can engage in these oscillations. And so the idea, so there are models that explained why this happens. So you can reduce, this is due to feedback inhibition from different neurons. But then the question is how do these local oscillations, how do they organize at the global level? And do they play a role in the brain rhythms that we detect at different frequencies? So to understand this, we employed, this was my PhD project with Gustavo, but since then I've been evolving on it for a long time. But the idea of Gustavo was to reduce the activity of a neural population to a phase oscillator. So something just oscillating at 40 hertz, like we discussed that 40 hertz is typical, already was addressed here. Now, if you have two local field potentials that are oscillating at 40 hertz, and they are coupled together, so there is diffusion between them, the Kuramoto model of coupled oscillators predicts that they will synchronize, right? So in time, maybe it can take some time, but they will eventually synchronize. But what if there is a conduction time? So how it takes time, so there is resistivity, so it's not immediate transmission. It's not like the speed of light. If this conduction time has a significant, let's say, it's non-negligible with respect to the frequency of oscillation, which is this omega n, or the period of this frequency, then we need to consider it in the equation. So when we are coupling the different units together, the different n to the other piece, we need to take into account how the activity was in the time delay before. And these are the different parameters of the equations, the coupling, the local connectivity. So they will synchronize. But another nice thing is that with time, the synchronization occurs at a reduced collective frequency. So what happens is that the frequency of synchronization, pointer is, sorry, wait, okay. the frequency of, this is important, sorry. So the natural frequency, the original frequency, or the average frequency between the two, is divided by 1 plus the coupling. And if this delay is larger than 0, and if n is very large, this collective frequency will be reduced significantly. If this is close to 0, it is usually neglected. So when we have many, let's say, oscillators, if they are uncoupled, they are all synchronized, let's say everything is segregated, there's no integration, everything is separated. Now, if you couple them together, these coupled oscillators in nature tend to synchronize together, right? And this Kuramoto model considers only the phase of the oscillations, disregards the amplitude, but is able to reproduce some phenomena that are observed in nature. And importantly, this global frequency, again, depends on the size of the system, so the number of units, and the speed at which the signal propagates. So this is relevant for these systems with large units and finite propagation speed. Now, another thing is that if you have complex network topologies, maybe the full synchronization is unstable, but you can have, you see here, this, I make these 4 that are more coupled together, they are synchronized, and these ones are desynchronized. And this dynamics, I cannot make more animations, but this is metastable. So a group can tend to synchronize and then desynchronize, then another subgroup can synchronize. And this induces metastability, so there's no full stability. The system is constantly trying to synchronize, but never fully synchronizing. There's a wide regime of that. And it induces frequency modulation over time. So if we look at the synchronization, let's say the global synchronization, like Gustavo was saying, if we take into account how the whole system, so here the global, like the global workspace is what characterizes everything. And then you have each unit, but that is part of the global, all of that. So if you increase the synchronization, let's say it's like more integration, and the synchronization over time can be fluctuating. So it goes up and down and up and down. And what this means, okay, so it's like you are fluctuating between segregation and integration. So there are these stories why these fluctuations are important. So when you are at the segregation level, so low synchrony, the oscillators are faster and out of phase. And when you are in periods of higher integration, everything synchronizes at slower frequencies, or cluster synchronize. So there is like an inverse relationship between synchrony and frequency, right? So this was the mechanism that we used then to try to see if this could explain the origin of fluctuations in the envelopes of MEG signals or EEG signals. So when we detect the signals from different brain areas, and what is considered functional connectivity, envelope functional connectivity, is when the power in a given frequency band is correlated. So this is observed in MEG, and then we made a model using these coupled oscillators, like the Kuramoto model of coupled oscillators, and we put them all at 40 Hertz. And when we filter in the same band as, so we find the right range of coupling and conduction delays, such then when we couple them together, we see this transient emergence of beta band oscillations, in this case, that are correlated across different areas. And this relates to the degree of synchrony. So again, the synchrony correlates to the power at different frequencies. But when we simulate with Kuramoto, what we are simulating is just a phase oscillator. So there's no change in the amplitudes, right? The amplitudes are constant. But if we take a look closer, it seems that sometimes the phases go faster, sometimes it goes slower. So you see more longer periods between two peaks or shorter periods. But the correlation, for example, here between these two signals is 0.06. They are not correlated. Now, if we filter these signals in a given frequency band, in a collective frequency band where I know that they were synchronizing, now the envelopes correlate 80%, right? So because here and here, at this instant of time, there was the short formation of a metastable synchronization, and they synchronize at a reduced frequency. And if we go to, a different frequency, and this is the frequency, the natural frequency of the oscillators, again, the correlation is 0. So there is this mechanism in the model, if this is the reality, of metastable synchronization that induces this power modulation of collective frequencies. So this is the frequency. And every time here, the synchrony degree increases a little bit. It's not full synchrony, but we see the emergence of a peak in the power spectrum that generates an envelope fluctuation. So now, but the thing is, how valid is this reduction? So the mechanism, okay, we explain it, but then in the brain, it's not really coupled oscillators. So the LFP gamma frequency oscillations are not in a limit cycle and oscillating forever. So they depend on decay time constants of neurotransmitter concentrations, and they emerge only briefly. So the idea is that these are so-called damped oscillations. So it's something more like this. This is the hop that we call the damped in the subcritical regime. So the oscillations don't last. So this is an example. So in the green case is when an oscillation is damped. So when we see those plots that we see, the limit cycle is when it's oscillating like this in phase space, and then in the phase portrait, let's say, and then when it's damped, it decays like this, and then it can be over damped as well, which is the red one, which is if it's really damped. Now, a system that it has damped oscillations, when it's perturbed with noise has all the possible frequencies, so it will resonate at its natural frequency with fluctuating amplitude, let's say. So you could imagine you have this cortical piece of tissue. The concentrations are of neurotransmitters and things that excite are subject to noise, thermodynamic fluctuations, let's say, and they transiently activate. Now, the more you can change some parameters in the equation to make the damping steeper or last longer. So it's like, you know, when you play a battery, like how long the oscillations last after you keep it at the pure frequency, right? Okay. So now the idea is, can this collective slow waves still emerge from interacting units that have a damped oscillatory response? So that was the idea, to use the Hopf model to see if we could still, so this is the equation. Okay. So this is the work that is now on archive. So this is the difference between what was addressed with the Kuramoto and with the Hopf now in the subcritical regime. So if the coupling is weak, the order parameter is low over time, the synchrony degree. This Kuramoto order parameter is COP. If the coupling is very high, you see here, the COP becomes stable close to 1, and you see these collective oscillations here at 2.5 hertz. So it's the same structure. All units only have a resonant frequency of 40 hertz, but they are coupled. And here in the intermediate coupling, you see that the degree of synchrony fluctuates over time. And this generates slow waves that are here in the middle at 7.5 hertz, for example, here. So it shows in the alpha band. And so then we do this analysis in parameter space. So the more we couple things together, so when we go up, the more synchronized the systems are, but the more time it takes between units, so the longer the conduction speed, it's harder to synchronize. So red means that it's synchronized, and this has been reported in the literature for a simple network. So this shape, that there's a high range of strong fluctuations. This is the metastability, strong fluctuations in the synchrony degree. Now, the peak frequency, so here this peak frequency here, that here is near 40 hertz, here 7.5. So these 3 points are the red, the crosses that we see here. And we see that the peak frequency, once the system reaches a moderate level of synchrony, we observe here this change, so red is 40 hertz, gamma. And when you increase the coupling, you start seeing that the peak frequency goes down to, can go down to alpha, down to delta, whatever. So this is the predicted global frequency from the equation, considering just all-to-all nearest neighbor coupling, very simple models of coupled oscillators. But it matches the analytic predictions. And then to find where in this parameter space are human brains, healthy human brains operating, if this could be a model. So we took the MEG power spectrum from, this is 89 individuals from the Connectome project. So they, on average, they have a peak here around the alpha band. And we find that where we have the best fit, let's say the less disparity between the mean MEG, real MEG power spectrum and the model is in the region of parameters where we had high metastability. So this is a bit how the power spectra look like in the parameter space. So in the region of metastability, there's more like a spectral entropy. So there are more frequencies appearing, the system is more rich. which I think maybe could also explain. So if we look then at the simulated time series, okay, so in all the 90 brain areas, and we filter them below 30 hertz, and here I'm coloring in blue or in yellow, in red or green, every time the power in that frequency range is exceeding 5 standard deviations above the baseline when no delays are included. So if you don't have delays, everything just synchronizes at 40 hertz and that's it. If you have time delays, when things synchronize, they synchronize at a slower frequency, and the power of this frequency really goes up compared to baseline. So we can detect, and here I'm going to zoom in, but still just to show. So this is the power below 30 hertz and the Kuramoto order parameter, and there's a correlation of 80%. So really, when you synchronize more, you have more power at lower frequencies. And just if we zoom in, just to show. So here, for example, you see that it's clear that we see these waves appearing. If there were no time delays, like you just see everything like noise, or for very weak coupling as well, we see this. Now, if we have very strong coupling, what happens? Then the stability of these oscillations, this collective oscillation, lasts longer, so this oscillation, and also they involve more units. So it's like everything is more synchronized for a longer time, and really at a slower frequency. So here the yellow is the delta range. So you can have 40 hertz oscillators that synchronize in the delta range. And again, the order parameter here synchronizes with power. So we characterize this so-called metastable oscillatory mode. So size is the number of units that simultaneously exhibit an increase in the power at a given frequency. And the duration is how long the power stays above 5 standard deviations. And then the occupancy is over the total, like the proportion of time that we detect these oscillations. And we find that according to these global model parameters, so the global coupling, for example, could be related to cholinergic transmission or the conduction time, could be related to global parameters that modulate the whole fluid, the fields, let's say. When Gustavo said that we don't have water in the brain, I'd like to counter-argue that 90% of our brain is water. But the thing is, like, there are different parameters that could modulate the conductivity of the fields, I don't know, during time, over time. Now, also, typically people look at the functional connectivity, so the correlation matrices between the envelope fluctuations. So if we take in the beta, so this is for weak coupling. All these functional connectivity matrices are very random, centered at 0, and the maximum is like 0.1. Now, in the optimal range, we find the maximum correlations between envelope fluctuations is in the alpha band, and it's mostly here at the superficial areas of the cortex. This depends a lot on the structural connectivity that we use. And then with strong coupling, you see here, like it's distributed over the whole cortex. There's strong strong delta. So this is the delta all over the place. They're not a single source, it's a collective phenomenon. If we, for example, increase the delays, there are also interesting phenomena that occur, and so there's a lot to explore in this sense. But one thing that I think is the take-home message, let's say, is you can have the same network structure, so a fixed network structure, and then just changing global parameters that could be like anesthesia or something that affects the whole field as a whole. It can affect the dynamics of the system or the regime in which the network is behaving. And maybe if we now perturb this model and then see how it reacts, probably in this region where we have more metastability, more the critical region, is where we would have more complex, like if we measure the perturbative complexity index or something like that, it's also probably the region where we have more turbulence. Whereas if you are in this region of strong coupling, if you perturb, like this perturbation will be highly synchronized and absorbed, and in the others maybe it will dissipate faster. So it could be interesting to see if different states of consciousness or if different behaviors. So this is Every time an unit has power above 5 standard deviations, we show a color. So in the intermediate range, every time you see blue, it's alpha emerging. But we didn't find that it's always restricted to the same areas. So it's very stochastic, let's say, I don't know, but maybe it depends a lot also on the number. We just use 90 units. So if we had a more realistic model, better connectivity matrix. So this is in the line. I always say that all models are wrong, even in the Kuramoto model. I'm not saying that the brain is the Kuramoto model or that neurons are not useful. Of course they are. It's just that if we are interested in understanding the mechanisms, the fundamental principles behind the things, we need reduction. Otherwise it's too complex, and the simulation results are again as complex as the real activity, and then we cannot compare with analytic works, those works that really solve the equations for simplified models. So we have the codes on GitHub, Francesca's GitHub. Francesca over there is the second author of the paper. And Jakub was, I will say this as well. Anyway, so one of the things is this model is not a predictive model. And as I say, when the reviewer says, but the fits too, no, this is more qualitative in the sense we just want to show that these things, so the locally generated gamma activity that you detect in vitro, maybe if you couple can generate larger, slower oscillations. And also, like once we have these constitutive equations or better models, we should be able to improve the prediction of the reaction of the system. And hopefully there will be more results coming soon. So this is work mainly with Francesca, Jakub, Martin, and Gustavo. Of course, Gustavo is the senior author of the paper and the inspiration, and we had the idea of using the Kuramoto to improve using the hop to improve from Kuramoto. So we first used the Wilson-Cowan models, the Apple oscillator. When I started my PhD, it was, can we even reduce this to Kuramoto to compare with the literature? Because there's a lot of literature on Kuramoto analytic solutions. And then we now increased again the realism with the amplitude fluctuation. And it's in collaboration with, Christian Bick and Renaud Lambiot are from the mathematics department in Oxford. So they helped in verifying the validity of this. And Vladimir Livtak is the supervisor, Francesca, expert in MEG. And Carl Friston, I think I don't need to introduce. And I'm funded by the Portuguese Foundation for Science and Technology. And thank you very much.